Measure histocompatibility, complex antigen. The definition, it's a glycoprotein antigens found on nucleated cells coded for by a complex of genes on a single chromosome, historically first recognized in 1940s in experiment studies on graft rejection. The nomenclature initials of species followed by LA, leukocytic antigen, human leukocytic antigen, mouse leukocytic antigen, like this. Foreignness is one of the essential requirements of a molecule to be immunogenic. The increased degree of foreignness of an immunogen is associated with increased intensity of immune response inside incitigated against it. When substance protein is injected into a host that have no protein identical to it, the injection protein, which is foreign to be immunized host, stimulate immune system for production of specific response. Foreignness, however, doesn't only means complete absence of identical molecule in the body of the immunized host, but also means structure difference of similar molecules. The following example makes this point clear. Insulin is a short polypeptide chain having molecular weight of 8,000 Dalton. It functions to control blood sugar level. Diabetes patients suffering from insulin deficiency can be treated with insulin prepared from human, bovine, swine, chicken source. Insulin types prepared from these animal species are similar in function and general structure, however, it differs in its molecular structure. There is difference in the sequence of three amino acids among the three types of insulin. Such difference can be recognized by the immune system, at, and it responds to the different types of insulin by production of antibodies specific to each insulin type. The history of measure histocompatibility. Transplantation, graft rejection, immune response, antibody formation, highly polymorphic, bind peptide recognized by T cells, three-dimensional structure determined by X-ray crystallography, a structure of the class 1 measure histocompatibility. This is measure histocompatibility class 1, and this is the alpha, beta 2, A3, and alloantigenic site. Here the membrane, plasma membrane, and cytoplasm. Structure: two polypeptide chain along alpha chain and a short beta chain called beta 2 microglobulin. Four regions: peptide binding region, a groove formed from alpha 1 alpha 2 domain of the alpha chain. Immunoglobulin-like region, highly conserved alpha 2 domain, site to which CD8 and T cell bind. Transmembrane region, stretch of hydrophobic amino acid, spanning membrane. Cytoplasmic region, containing site for phosphorylation and binding to cytoskeleton. Structure: peptide binding region, a groove composed of an alpha helix on two opposite wall and 8 beta plated sheet forming the floor. Reside lining groove most polymorphic. Peptide in groove 8 to 10 amino acid long. Specific amino acid on peptide required for anchor site in groove. Variability of polymorphism. Structure of class 2. Here a NH2, NH2. Two polypeptide chain alpha and beta of roughly equal length. Four regions. peptide binding region, a groove formed from alpha 1 beta 1 domain of the alpha and the beta chain site of polymorphism. Immunoglobulin like region conserved alpha 2 beta 2 domain. Beta 2 is site to which CD4 on T cell binds. Transmembrane region stretch of the hydrophobic amino acid spanning membrane. Cytoplasmic region contains site for phosphorylation and binding to cytoskeletal elements. Peptide binding groove for class 1 and class 2 major histocompatibility are structurally similar. Both have a peptide binding groove with a wall of 2 helix and a floor of 8 beta plated sheet. Close-ended groove for class 1 major histocompatibility require an 8 to 10 amino acid length peptide to bind. Open-ended groove for class 2 major histocompatibility lets it bind a peptide 13 to 25 amino acid long, not all of which lie in the group. Anchor site rules apply to both classes. Aspect of major histocompatibility. Major histocompatibility molecules are membrane bound. Recognition by T cells required cell-cell contact. Peptide from cytosol associated with class 1 major histocompatibility and is recognized by TC cells. Peptide from vesicle associated with class 2 major histocompatibility as recognized by THC. MHC. Although there is a high degree of polymorphism for a species, an individual has maximum of 6 different class 1 MHC product and only slightly more class 2 MHC product. A peptide must associate with a given MHC of that individual, otherwise no immune response can occur. That is one level of control. Mature T cells must have T cell receptor that recognize the peptide associated with MHC. This is the second level of control. Each major MHC molecule has only one binding site. The different peptide a given MHC molecule can bind to the same site, but only one at a time. MHC polymorphism is determined only in germline. There are no recombination mechanism for generating diversity. Because each MHC molecule can bind many different peptide, binding is termed degenerate. Cytokine, especially interferon alpha, increase level of expression of MHC. Alleles of MHC genes are codominate. Each MHC gene product is expressed on the cell surface of an individual nucleated cell. Why the high degree of polymorphism? Survival of this species. Structure of T cell receptor. Alpha chain, beta chain, variable region, and constant region, high H transmembrane region. Structure. Two polypeptide chain, alpha and beta, of roughly equal length. Both chain consists of variable and a constant region. Alpha chain V region has adjoining segment. Beta chain V region has both G and diversity D segment. Hypervariability region in V contribute to diversity of TCR. TCR recognize T cell receptor recognize portions of MHC molecule and peptide bound in the groove. Small population of T cells has TCR comprised of alpha and beta chain, upsilon chain, specific different from alpha beta TCR. This is the structure alpha and beta chain of T cell receptor. Properties of Ig and T cell gene. Many VDG, few CS. Ig and TCR, VDG rearrangement, V pair from antigen recognition site, somatic hyperimmunization, transmembrane form, secretory form, isotype with different functions, valency, CD3 complex group of 4 protein associated with TCR consists of alpha, gamma. All 4 proteins are invariant. Function synthesized coordinately with TCR required to bring TCR to surface, transduce activating signal to T cells when T cell recognize MHC peptides, accessory molecule involved in cell-cell interactions, T cell surface molecules that engage with ligand on second cell when TCR recognize MHC peptide, T cell ligand of 2 second cell, CD4, CD8, LFA, LFA3, leukocyte function associated antigen, intercellular adhesion molecule, all are invariant, increased adhesion between two engaged cells. Some show increased expression in response to cytokines, co-stimulatory molecules on T cell and second cell that engage to deliver signal required for activation of T cells. Most important co-stimulatory molecules, T cell ligand and CD28, interaction of T cell and the antigen presenting cell. This is the antigen presenting cell and this is the interleukin 1, interleukin 6, tumor necrosis factor alpha, interleukin 12, interleukin 15. This is other factors. T cytotoxic lymphocyte, interaction of T cell and